Yeah, hello and a very warm welcome also from my side. And yeah, thank you for joining our today's webinar, Thinking Translational, Things to Consider in CAR-T Research Workflows. Please briefly acknowledge also our disclaimer displayed here on this slide. Taking a look at our today's agenda, we firstly take a look at the path to cell therapies from early discovery to commercialization and the challenges and needs that you might face in the different phases. Then I will elaborate the key points to consider to ease up the transition from small scale to large scale manufacturing. And then I will also talk about suitable tools that we have been developing that enable a smooth transition from the small scale research setting to the large scale manufacturing workflow, and of course, end up with a summary and some links and resources. I'd like to start off with a question. So, what's your vision when it comes to CAR T cell therapy? I'm sure that every one of us has his or her personal goals, but beside that, the overall vision is to develop CAR Ts that work, translate them into the clinics, and successfully treat cancer patients all over the world. However, as we all know, it's quite a long way, starting from basic research and discovery to preclinical development to the different clinical phases to a marketed therapy that really makes a difference in patients' life. And especially cell and gene therapies come along with a lot of hurdles and challenges, which is especially true for such novel therapeutic approaches using such living drugs like CAR T cells. And of course, the challenges differ from phase to phase. In a research setting, you need to narrow down targets, develop safe CAR T cells, validate them in vivo and in vitro. In the translational phase, you need to transfer your small-scale protocol to a large-scale process, and later on, you also need to take care of regulatory requirements, deal with increasing numbers of patients, and of course, always keep an eye on the costs. And of course, although the needs will change, at the discovery phase, you need a high degree of flexibility and a diverse set of methods. Whereas later on, standardization and process automation will be key. And by this, it becomes obvious that the path to CAR T cell therapy is not an easy one. However, it's doable, as reflected by the increasing number of commercial CAR T-cell products. And by today, already 6 therapies are marketed to treat CD19 and BCMA-positive malignancies. And a lot more are in the pipeline. By today, a total of 1,115 clinical trials are initiated, and the numbers are rapidly increasing, as shown by this graph here on the right-hand side. The drugs are currently tested in clinical studies worldwide, underwent the depicted path, and have had a need for phase-specific solutions, and for solutions that enable a transition from one phase to the other. And as Miltenyi is very active in the field of cell and gene therapy, we also experienced such challenges and hurdles and developed integrated workflow solutions and tools, such as a research workflow, translational research workflow, as well as clinical workflow and patient monitoring workflows. But now you may ask, what distinguishes Miltenyi from all the other solution providers? So, well, our corporate mission is to advance scientific research in medicine by providing solutions for curative cell and gene therapy as well as biomedical research. And this mission is the backbone of the company and influences our daily work, from research to clinical, from R&D to marketing. And given our maxim, Make Cancer History, Miltenyi is very active in the field of cell and gene therapy. Our R&D department is working on the development of CAR therapies, reflected by several scientific publications, conference contributions, as well as many collaborations with academic labs and consortia, for example, the CARAT or the T2Evolve consortium. And this also led to a proven track record of our solutions by scientific publications in high impact journals and clinical CAR T cell trials. And given our great expertise and experience in the CAR-T research as well as in the manufacturing field, we want to share now within the next chapter some of the considerations to streamline the transition from the small-scale research setting to the large-scale clinical manufacturing of CAR-T cells. First of all, it's always helpful to know right from the start where the journey is going to. And thinking translational, as this webinar of today is named, pays off at the end. It not only saves time, but also may reduce costs, and it's easy on the nerves. So taking a look at the reagents, for example, here the questions should be, are my reagents available in GMP quality? Are this the same clones and formulation, or does this differ? And how does this influence my process? Also, in terms of their packing, is in a large scale, is it then bags versus flasks in small scale? What is about the supply chains? Are my reagents available worldwide? Are they stable at room temperature? If I produce in a cell factory, cell factory environment, what is about my antibodies that I am using in my panels? Are they really reproducible? What is about my in-process control? Do I already have suitable antibody panels, or do I need to re-establish them when I switch from small-scale research to a large-scale manufacturing? If it comes to the device, I will need for sure automation. What is about the scalability? Is the process then easy to scale up? Is there a proven track record? Does the community uses this device? What is about the robustness? And also, what's also super important is the technical support. What is if something is happening? Do I have technical support on my device? Then in terms of the methods, are the methods translatable to an automated system? What is about the time of the assay? Can I shorten it? Because time in the end is also influences the cost. Are my methods really reliable? How many readouts do I need and do I want to have? Does my method fit the purpose? Is the assay suitable for a QC in a clinical setting? And are those assays already used in clinical trials? And all those considerations, if you put some thought in it up front, this will really streamline the transition from small scale research to large scale manufacturing. Yeah, we follow the workflow depicted here on this slide, starting from human blood products, followed by CD4, CD8 T cell isolation, the activation of the T cells via CD3, CD28 receptor engagement, followed by gene delivery by viral transduction or electroporation, then the ex vivo expansion of the CAR T cells, as well as a phenotypic and functional verification at the end of the workflow in a research setting, and this is also quality control in a clinical setting of the final product. And we integrated and developed a broad range of products that are used in research and clinical setting. And yeah, using such products already in the research phase enables for a seamless transition, or allows for a seamless transition of your whole research workflow to the Clinimax Prodigy platform, where GMP compliant CAR T cells can be manufactured automatically and in a closed system. Automating and closing of manual steps makes manufacturing more scalable, robust, reliable, and consistent to enhance the product quality. And yeah, human error is frequently identified as one of the highest risk elements of the manufacturing process, leading to protocol deviations and batch failures. So automation mitigates the risks by providing repeatable and reliable cell processing. And keeping that in mind, I would like to introduce our Clinimax Prodigy platform to you. For those of you who do not know it now, by now, this is our hands-off end-to-end process platform. And I just want to quickly introduce the key features to you. So it is an integrated and flexible platform with one closed consumable and a reduced footprint, so it's not too big. And it's also fully automated. It's a fully automated platform that absolutely increases the reproducibility and standardization. It's a closed system, but with an in-process control and quality control sampling that you can increase the robustness of your process. It comes along with a flexible software and parameterization. And what is also really nice is that the Clinimax Prodigy platform is used in more than 180 clinical trials worldwide so far. Yeah, as I just mentioned, the Clinimax Prodigy platform is very flexible and comes along with a lot of processes. And one process is the T-cell transduction process, in short, the TCT process, which includes the C-cell separation, activation, transduction, and expansion, as previously mentioned. And yeah, it is for the automated manufacturing of CAR T cells, starting from different blood products. There are 2 enrichment strategies available. You can customize your culture conditions, and you, of course, can do a retroviral and lentiviral transduction strategies. Yeah, especially for the TCT process, we have a proven track record in T-cell engineering, and yeah, from small-scale research to large-scale manufacturing with our Clinimax Prodigy, and from 2016 on to 2022 now, we have a lot of preclinical data and manufacturing optimization, different publication in peer-reviewed journals, as well as first clinical results. I do not go into detail, but this is, yeah, the customer experience regarding this. Yeah, furthermore, the TCT process, the T-cell transduction process on the Clinimax Prodigy as an application is already used in several clinical trials worldwide to manufacture CAR T cells. Yeah, so after this short excursus to the to the Clinimax Prodigy and the T-cell transduction process for the clinical manufacturing, we are now coming back to the translational research workflow, and we want to take a closer look at the workflow itself and look at what is important to consider to ease up the transition from the small-scale research setting to the large-scale manufacturing to shorten the development time. Yeah, depicted here on this slide, you can see the translational research workflow, so the experimental time scheme of it. And yeah, I will just let you through it. So we started with the T-cell isolation on day 0, followed by the stimulation of the isolated T cells. We also did an immune cell composition analysis by flow of the isolated T cells before and after enrichment. We also checked for the activation prior to activation, and the activation was also checked throughout the whole procedure. We performed a gene delivery on day 1 after activation. We washed away our activation reagent on day 3, and we also used flow analysis panels to assess the stem memory T cell phenotype. And of course, after activation, the expansion period took place until day 22. Yeah, looking at the first step, the T-cell isolation. In a research setting, several methods are frequently used. Some start directly with PBMC, so no enrichment at all. Others go for pan-T-cell isolation, or others go for positive selection, for example, of CD4 and CD8 T-cells. And when it comes to translation, there are some questions, some considerations coming up, namely, the optimal reproducibility of the results by the different methods, by the different approaches. Are the reagents available in GMP quality? So, for example, pan-T-cell isolation kit or CD4, CD8 microbeads. Is the isolation method translatable to an automated system? So if you go for PBMCs, is this translatable? And are the clinical processes already established or available? So is there a proven track record? Is someone already doing it? Yeah, so we always go for CD4, CD8 positive selection, and yeah, I will also let you know why this is beneficial for the whole process. And so we assess the immune cell composition using a predefined antibody panel before and after the T cell isolation with CD4, CD8 microbeads. And you can see the numbers of the three different donors. And these graphs nicely show the heterogeneity of the in the cell composition in the starting material, here Buffy codes, and this heterogeneity would be even more obvious if we would use a patient material. But however, after the enrichment, the amount of CD4 and CD8 T cells between the different donors was comparable. And this means that we start with a more comparable and a more homogeneous sample, which then clearly leads to a higher reproducibility of the whole process. Yeah, additionally, we observed a correlation between the numbers of monocytes in the starting material and the transduction efficiency. And the hypothesis here is that the monocytes eat up the virus, and as shown on the figure on the right-hand side here. And this also leads to the conclusion that pre-enrichment is also beneficial for the transduction efficiency, to improve the transduction efficiency. Yeah, besides the higher reproducibility and the improved transduction efficiency, if we remember the considerations, the CD4, CD8 microbeads are also available so they are available in research grade as well as in max GMP quality. The positive isolation is compatible with the Clinimax Prodigy T cell transduction and T cell TCE process for the T cell electroporation, and our max microbeads are already used in clinical trials worldwide. If you find yourself in a research setting still working with research products, we have two options available that I want to highlight here. One option is to isolate PBMCs from blood products and continue with a one-to-one mixture of CD4 and CD8 microbeads, including the cycle. This will take approximately 2 hours. And yeah, as time is money, we are using our straight from microbeads frequently, and those straight from microbeads, are added directly to the blood product. And by this, you can process, for example, an entire Buffy code within only 30 minutes. And yeah, both are positive selection approaches and thereby are translatable to a large scale manufacturing at the Prodigy, and they are 100% comparable. So in more detail, on day one, the T cells of each donor were transduced with equal amounts. On day one, T cells of each donor were transduced with equal amounts of lentiviral particles encoding a CD20-directed CAR, and the transduction efficiency was analyzed by flow cytometry using a CD20 CAR detection reagent, anti-human, and independently of the manufacturing protocol. So if you go for standard straight from microbeads or the standard microbeads, we observed a comparable transduction efficiencies on day 6 as well as on day 12. There were no significant difference. We had to assess and compare the cytotoxic or cytolytic potential of the CD20-directed CAR T cells. CD20 positive JACO1 cells were co-cultured with CAR Ts derived from two different protocols at different effector to target cell ratios for in total 20 hours. And yeah, analyzing both target cell lysis as well as the release of pro-inflammatory cytokine like GM-CSF, interferon gamma, IL-2, and TNF alpha, we could confirm that there's no significant difference in regards to cytotoxic potential of both cellular products. So by this, we could confirm that there's no difference between the straight from microbeads and the standard microbeads, but the straight from microbeads are, of course, the faster option. Yeah, independent of the approach you decide for standard microbeads or the straight from CD4, CD8 microbeads, the basis is always our MAX technology. But why is it so beneficial to use the MAX technologies for CAR T cell engineering, for T cell engineering, immune cell manufacturing? So our technology is based on the MAX columns. And through the columns, the magnetic field can be or is amplified by a 10,000 fold. And this allows us to reduce the number of beads of these nano-sized max microbeads, and this leads then to a minimal labeling of the cells. And further, there is a large space between the steel spheres of the column matrix, and that enables a free flow through of all the non-labeled cells and an enrichment of the minimal labeled cells without any unspecific activation. And yeah, if we take a look at the electron microscopy images here at this side, comparing a column-free system versus our MAX technology, you can clearly see the bead aggregates on the surface of the cells if you go for a column-free system, whereas cells isolated with the MAX technology, due to the minimal labeling, look really, really natural and healthy. And yeah, taking all these benefits into account, it becomes clear why our MAX technology, our column-based technology, is already used in clinical cell therapy trials worldwide. Now, as we have enriched our T cells with the straight from microbeads or the CD4, CD8 standard microbeads, we can continue with the stimulation of our isolated T cells, and also performing flow analysis to check for the activation of the T cells. For this workflow step, the physiological activation of T cells and then the efficient expansion, there are also several options available. So T cell activation via a nanomatrix, a bead-based T cell activation, or also functional grade antibodies to perform the CD3, CD28 stimulation of the T cell. Yeah, what are the key considerations for this workflow step? So are my results reproducible with my activation method? Are the reagents available in GMP quality? Are the activation reagents suitable to an automated closed system? Do I have control of it? And is a clinical process already established and available? Is there a proven track record of these reagents, clinical phase? And also, what is about the safety of these reagents? We considered such things, and we meet the needs and developed a product. This is named T-Cell TransAct, and this meant for physiological, or was developed or has been developed for the physiological activation of T cells and for their efficient expansion. And what makes this reagent special, so it's a colloidal polymeric nanomatrix that is conjugated to CD3 and CD28 agonists. It can be dosed volumetrically, and it can be removed by a simple wash step. So there's no need to add any chemicals or a bead removal step. And it is physiological, it enables a physiological and robust stimulation of the enriched T cells, in our case CD4, CD8, and it's also suitable for an automated manufacturing platform due to these features as volumetric dosage and removal by a wash step due to this nanomatrix. And it's also available in research grade as well as in max GMP quality, and TransAct is also already used in clinical CAR-T trials worldwide. Yeah, procedure-wise, the T cells were pelleted and were suspended at a concentration of 10 to the power of 6 cells per ml in Tex-Mex medium, supplemented with our T-cell TransAct and cytokines. So here we used IL-7 and IL-15. And from this cell suspension, 2 ml were filled into a 24 well plate and incubated for 24 hours. And 24 hours after seeding, the T cell, the gene transfer was done. And yeah, 3 days after the activation medium was replaced by Tex-Mex supplemented with IL-7 and IL-15. Gene modified cells were split at least every third day, or when the cells, of course, are confluent or exceeded a confluence of 70%, they were also split. And then we did flow analysis on day 3, 6, 8, 13, 15, and 17. using the T-cell activation panel consisting of CD3, CD4, CD8, CD25, CD69, and CD137. Applying the T-cell activation panel, including the markers CD3, CD4, CD8, CD25, CD69, and CD137, we observed that all three markers were upregulated upon T-cell activation using T-cell TransAct via TCR signaling, but with different kinetics. And CD137 and CD69, they are very early activation markers, and they are usually detectable within hours after the T-cell receptor ligation, and the expression is induced. And after 48 to 60 to 96 hours, the expression then decreases and goes back to the baseline, as shown here. Yeah, for the other marker, for CD25, this is the alpha chain of the IL-2 receptor, and this is upregulated a bit later usually than CD69 and CD137. But it's also detectable on day one. The data are not shown here, but what is shown here is that the CD25 expression remains high out to day 8 to 9, and then starts to go down after day 8. Yeah, after the expansion period of 14 days, we applied T-cell TransAct again to induce or to see if the T-cells can be re-stimulated. And what we've seen is that there was again an upregulation of CD137 as well as CD69 and CD25. Yeah, additionally to the data I've shown, I also want to highlight the safety aspect of T-cell TransAct, of the Max GMP T-cell TransAct, because using immunomodulatory substances for the manufacturing of cellular products for example, advanced T-cell therapies, a safety assessment of the reagent that I used is essential to identify potential risks associated with or to their use. And yeah, with several experiments, we demonstrated the safe use of the Max GMP T-cell TransAct on the automated Clinimax Prodigy platform using the previously mentioned TCT application software. And I don't want to go into detail, but highlight that T-cell TransAct is really a robust and potent polyclonal T-cell activation and expansion tool for clinical scale manufacturing of modified T-cells. And if you're interested in more details in the data, then you can scan the QR code here, and this will lead you to the poster. Yeah, next you would like to take a look at the expansion of our transduced or electroporated T cells. So how can we ensure a highly efficient expansion? And here as well, here are basically two options. So of course you need medium and cytokines, but you can go for medium without serum, or a serum-free medium and a cytokine cocktail. And also in this T-cell expansion process step, there are several considerations. So, for example, are my cell culture results really reproducible, and how can I ensure this? As well as, are my reagents again available in GMP quality and in research quality? Is my medium available with and without phenol red if I need it, if I would like to have it? What is if I switch from bottles to bags, so from flask to bags, from research to a clinical setting? And what is about the unique specific dosing? And yeah, after successful transduction, the T cells were expanded, as said, and this was done in serum-free Tex-Mex medium and max cytokines IL-7 and IL-15. And we went for especially for the clinical application for a serum-free system, which is advantageous because sourcing large amounts of quality AB serum can be very challenging or difficult. And for the clinical CAR T-cell manufacturing, we developed this serum-free GMP expansion to be used with Tex-Mex medium. And as depicted here, various starting materials, so healthy donors, patient material, as well as cryopreserved, so frozen patient material was used, and there was no effect on the cell count, as well as on the expansion and on the viability, also shown here for cell count and for cell viability. And yeah, especially what I wanted to highlight is that also for the frozen patient material, we observed really high viabilities and expansion rates following this workflow and cultivation with serum-free medium. The medium as well as the cytokines are available in research grade and in max GMP grade. And yeah, especially for the research, it's so not only especially, but for the research, our cytokines, our premium grade cytokines, that you also get a certificate of analysis and can make sure that you have a unit-specific dosing to ensure a really high reproducibility and then also an easy translation. Yeah, now I would like to take a look at the last step of the workflow, the phenotypic and functional analysis. In a clinical setting, in a manufacturing setting, this is then the in-process control, IPC, and quality control, QC, but of course you also do it in a research setting, but here it's not named IPC, QC, but it's basically the same. You check your cells, and there are also different options, so you can do it via flow cytometry, but there are also others. You can do omic analysis. Some do maybe also stainings or real-time qPCR, and there are a lot of more methods to phenotypically investigate or assess the functionality of the CAR T cells. And again, here are also things that you should consider. So in terms of the reproducibility of your flow cytometry results, also taking a look at the reagents. Is the panel performance confirmed? What is about the instrument harmonization? What is about the analysis? Is this user dependent? For sure, but how can this be done user independent? What is about the reporting? What is about the standardization of the protocols, and so on? And yeah, here we collected some main sources of variation in flow cytometry. And based on this, we at Miltenyi, we established a flow cytometry standardization concept. And there are different sources of variation. So for example, the donor variability, this is patient-driven, pre-treatment-driven, and also collection-driven. And there is not much you can do, but there are other things, for example, the reagents, to ensure a lot-to-lot consistency of your antibodies. Also for instrumentation, there is a pipetting variability, manufacturing tolerance, and operational tolerances. Then regarding the analysis and reporting, this is mainly user dependent and prone to human error. And also in terms of protocol, there are often non-optimized procedure and a lot of manual handling steps, which lead to a lot of variation in the flow cytometry analysis results. Yeah, what is depicted here is our flow cytometry standardization concept. And yeah, the main source of variation can be basically mitigated by our reaffinity antibodies. Those are derived from a defined DNA sequence, and they are produced under standardized conditions and provide a high lot-to-lot consistency. And what's also very nice is that there's only one IgG isotype, so there's only one isotype control needed for the experimental setup, and also there's no Fc blocking receptor step or reagent needed. And all the reagents are for IPC-QC are also available in clinical quality. So this makes it easy to transfer your panels from the research setting into the clinical setting and continue there. But not only the reagent, but also the instruments are made to create reliable and reproducible flow data. And with the so-called smart gain technology, you can take an instrument setting from one device to another and achieve equivalent performance. And the idea is to provide really reliable data from test to test, from user to user, and also from instrument to instrument across different sites. And this is also super interesting if it then comes to the clinical manufacturing. Then we also have our express modes. And this is a software add-on for our Max Quant analyzer software. And this gating adjustment is based on an algorithm, and this can be used to avoid variations caused by different humans, by different operators during the data analysis. And in terms of protocols, we also go for a high degree of automation, customization if needed, and we have a great application support. So if you would like to learn more about our flow cytometry standardization concept, we have a really nice analytics day, a round table, a lot of resources, and some really nice webinars that elaborate a bit more on these flow cytometry standardization concepts. So feel free to follow, to scan this QR code here and learn more. Now I come to the last part of this webinar, the summary and resources. Yeah, I hope that it became clear why thinking translational right from the beginning is really beneficial to streamline the transition from the research phase to the preclinical development. And that at least some of the considerations I mentioned at the beginning and during this webinar will lead to discussions about your current workflow, to adaptations, and that you can streamline and improve your workflow, and by this also save some resources, time-wise, money-wise, and also your nerves. On this slide, I summarized our solution for the research part as well as for the clinical manufacturing part. And as you can see, for every research product I mentioned in this workflow, the GMP counterpart exists, and this is developed, our solutions are developed to enable a smooth transition from the research phase to the preclinical development. I also compiled here some of the resources that I would like to share with you. So the first is our small-scale TCT protocol. This has been developed in cooperation with our R&D, or it's an R&D protocol basically that they shared, and it is a step-by-step protocol. It includes all the steps, all the material, all the tips and tricks to engineer CAR T cells, and already some of the customers are using it, and I always get very positive feedback. So take a look, download it, and it's also for free. Then we have also our application page where you find different product solutions, but also webinars to different CAR T topics. You find application notes, tag sheets, and a lot of helpful tips and tricks. And a bit old school, but still digital, is our CAR T cell research and discovery brochure, which also summarizes nicely all the tools and product that we offer to support your research. If you have any questions or you want to receive a deeper and more extensive information or personal consulting about our solutions, I invite you to contact our local sales organizations. And Miltenyi Biotec is represented in over 23 countries worldwide, and we also have representatives in a total of 73 countries worldwide. So please feel free to reach out, to get in contact, and to get more information. I also want to invite you to follow us on social media, LinkedIn or ResearchGate, and stay tuned and don't miss our latest scientific publications, free research protocols, travel awards, and also our latest product releases. Yeah, and with this nice image of our headquarter campus in Bergisch Gladbach, Germany at night, I finish my presentation and I thank you very much for joining this webinar today.